Welcome to Cellular Bioluminescence Imaging of Circadian Clocks, a webinar sponsored by Andor Technology. I'm Robin Riley, the web editor at Photonics Media. Before we begin, just a couple of quick technical notes. If you have any problems, the easiest thing to do is simply to log out and log back in to rejoin the webinar. A recording will be available shortly on photonics.com webinars if you miss any part of the presentation. I'll introduce our speakers in just a moment, but first a few words about Photonics Media. We publish the magazines Photonics Spectra, Biophotonics, Industrial Photonics, and Europhotonics. Subscriptions are free and forms are available on our website, photonics.com. During the webinar, you may type any questions you have in the question box in the webinar program, and we'll answer as many as time allows after the presentation. Please note that although we will be taking your questions throughout the hour, we will only answer them after the presentation is complete. Our speakers today are David K. Welch, MD, an Associate Professor at the UC San Diego Department of Psychiatry, and a staff psychiatrist at the VA San Diego Healthcare System, and Colin Coates, a product manager at Andor Technology, where he is responsible for camera technologies. David earned his undergraduate degree at Stanford University and received a PhD in neurobiology and an MD from Harvard University. Colin earned his PhD at Queen's University in Belfast, Ireland. Welcome, David and Colin. Please begin.
All right, this is David beginning here. Hello, everyone. Today, I'd like to tell you about our use of bioluminescence imaging to study circadian clocks. Now, circadian clocks are found in individual cells, and what they are is transcriptional negative feedback loops within these individual cells. So I have a little movie to show you what it is that we're studying in a cartoon form here. So we start out with let's see, we can start out with that. And of course, I'm missing the movie. I don't know why, but I'll take a look and see if I can find it for you. So while I'm looking, let me just tell you what the clock is all about. It's a transcriptional negative feedback loop, and I'm going to show you this application. Hopefully you'll be able to see that. Sorry about this. I really did test everything before beginning, but you never know. It's going to happen over the weekend. Okay. But I think this will be worth it because this is a very nice movie. Okay. So so here we have back to our cell and the circadian clock in individual cell. So BMAL and clock are a dimer. They form a dimer, and they activate the transcription of a family of period and cryptochrome genes, periods and cries, or PERs and cries. The PERs and cries are transcribed, translated, and form heterodimers, which then go back into the nucleus. And the key thing is that the complexes of PERs and cries ultimately inhibit this activation of their own transcription. Okay, so it's a delayed negative feedback loop made up of this transcriptional process. And the net result is that the PER and cry levels go up and up and up until eventually the inhibition builds up and they inhibit their own transcription. And then the levels go down as the proteins are turned over. And eventually the inhibition is relieved, and then the levels can come back up again. Okay, so let's go back to the presentation. Sorry about that. And so we take advantage of this window on the circadian clock in individual cells, and the fact that the components of the clock itself are rhythmic by using optical reporters to study the clock. So this shows you that both the RNA levels and the protein levels of the clock components are highly rhythmic with this approximately 24-hour rhythm. The function of the circadian clock, I should have mentioned, is to keep track of time of day for within individual cells and to organize physiology and behavior at the level of the whole organism according to time of day. And it all comes down to these transcriptional negative feedback loops in individual cells, which we can then measure at the level of individual cells with optical reporters. And the first such reporter that was developed was using the promoter for one of these clock genes per1 or period 1 to drive rhythmic expression of green fluorescent protein, GFP. And here you see in the upper left a fluorescence picture, still picture of the suprachiasmatic nucleus, which is the brain's master circadian clock. And below, there are 3 panels showing rhythms of fluorescence in individual cells from the SCN using this reporter. And I think I have a movie to show you. Yes, okay, that one works fine. So this is a time-lapse movie of SCN neurons glowing from the GFP driven by the rhythmic period 1 promoter. Okay. Now, fluorescence, I'm sure you're all familiar with, fluorescent probes are used very commonly in biology. The process is that photons from an external source are used to excite electrons in the sample to an excited state, and then these excited state electrons relax down to a ground state and in the process emit photons at a different wavelength. The advantage of fluorescent reporters is mainly brightness. Because you're bombarding the sample with a lot of incident photons, you get a lot of photons out. And because of the brightness, you get great spatial resolution, temporal resolution, contrast, and also because you can just use very short exposures and flip on the lamp to see individual cells. It's very easy to target cells for other studies, like in this picture in the lower right, showing that you can do patch clamp recordings of an SCN neuron, for example, just by flipping on a fluorescent lamp to identify which one is bright. Now, there's some limitations of fluorescent reporters that all come from the use of such a high level of incident illumination. There's photobleaching, phototoxicity. You can perturb the physiology of the sample, for instance, if it's the retina. And also, there's a very high background that comes from both the excitation light itself as well as autofluorescence that's nonspecific. So I'd like to introduce to you an alternative bioluminescent reporters in which it's a slightly different process. On the left here you can see that you still have an excited state of electrons, but to get to that excited state, instead of exciting the electrons with incident illumination, it's a chemical reaction catalyzed by luciferase that excites the electron, and then you have relaxing back to a ground state just like in fluorescence and emitting a photon. But because the energy comes at a much lower level and comes from a chemical reaction, you don't have the same kind of photobleaching, phototoxicity, perturbation of physiology that you would get with fluorescence. And the process is actually quite a bit more sensitive due to low background because there are no excitation photons, virtually no autoluminescence of a sample, and it's linear over a huge range. There's some limitations. You have to add the substrate for the enzyme, which is luciferin. And The main limitation of bioluminescence is it's really exceedingly dim. So you have no idea how dim this is compared to fluorescence. Consequently, the spatial and temporal resolution and the contrast are really not that great compared to fluorescence. Bioluminescence, just to get the terminology clear, is widespread. It's a naturally occurring phenomenon in nature, and luciferases are the enzymes that catalyze the emission of light from a substrate without requiring any exogenous illumination, whereas the luciferins are the substrates of the luciferases. And here is the most commonly used luciferase in nature. It's a firefly, lightning bugs. You might have seen these yourself. And the structure of the luciferase enzyme is known. This is the 3D structure over here. And there's a little movie with some background music, so you can get in the mood here. The firefly is lighting up. And over here on the right is the chemical reaction. Luciferin substrate is oxidized in the presence of ATP and emits a photon in the process. In our experiments, the probe that we used is actually within a knock-in mouse that was engineered in Joe Takahashi's lab. In this mouse, the endogenous PER2 gene, which is highly rhythmic, and it's what we want to measure, is replaced by PER2 with firefly luciferase, so that a fusion protein is expressed. And so everywhere PER2 goes, this firefly luciferase enzyme goes as well, and it reflects both the transcriptional and any kind of post-transcriptional regulation of the PER2 gene. So it's a very faithful reporter of what the circadian clock is doing in individual cells. And so the easiest way to measure this is to just not do any imaging at all, just to put the sample in a luminometer with a photomultiplier tube. You don't get any spatial resolution from this, but you can record from individual samples of, for example, an SCN slice and get these very nice rhythms of bioluminescence from a single sample. So either from, say, a tissue slice or a population of cells. But what we'd really like to do is to measure the circadian clock's function with single cell resolution, either cells within a tissue section or dispersed cells in a slice. And so to do this, we put the sample on a microscope, surround it with an environmental chamber or incubator, and use a low noise CCD camera. And let me show you some examples of time-lapse videos that we can get from these either SCN slices or dispersed SCN neurons from the knock-in mouse that I told you about. So this is the PER2 luciferase mouse that's going to let us look at the circadian clock by means of following these the glowing of individual cells over time. And so on the left, I'm going to play this movie, which is an SCN slice, and what you'll see is the cells within this tissue section are more or less synchronized. Get brighter and dimmer and brighter and dimmer. And every peak of brightness is about another 24 hours in this time-lapse video. Okay. And over here on the right is a culture of dispersed SCN neurons. And the difference here is that the cells are no longer connected with sufficient synaptic connections to maintain synchronization among individual cells. And so what we'll see is that the individual cells are still rhythmic, but they're peaking at different times. So, and what in this movie, the bottom panel isn't going to be an animated plot of the brightness of that one cell in the middle to which the yellow arrow is pointing. And so I'll show you that right now. Okay. So if you follow along here, you'll see that all these bright spots in the image are individual cells, and they get brighter and dimmer and brighter and dimmer, but they don't all get bright at the same time like they did in the slice, in the text slice. Okay, and this is a time lapse over a period of 2 weeks actually on the microscope, and it's a nice demonstration that individual cells are independent circadian oscillators, as revealed by this bioluminescence that is rhythmic. Okay. You can do this with other cells besides cells from the master circadian pacemaker, because circadian clocks are widespread. And here is data from fibroblasts from the same HER2-LUC mouse. So they're, each one of these plots is in individual cells. There are 8 different fibroblasts shown here. The rhythms of HER2 bioluminescence are plotted, and then on the right this is a movie. And this is just to show you that very, very long-term experiments are possible with bioluminescence imaging. And so this is, I think, the world record. It's a 6-week experiment on the microscope stage with fibroblasts. And the spots that the bright spots here are individual fibroblasts that wink on and off, kind of like Christmas lights. And they keep going and remain rhythmic for the whole 6 weeks of the experiment. Okay, I won't wait the whole time, even though it is speeded up. All right, now for the second part of the talk, I want to get more specific and practical and focus on things that will help you to actually do this, use this technique. I want to mention some things about reporter design, luciferases, lenses, cameras, optimizing settings, and image processing and data analysis. Okay. So first, reporter design. I should mention that although we love the HER2-LUC knock-in mouse as a bioluminescent probe, one disadvantage is that it occurs in only a single copy number per cell, so it's quite dim. And there are other options that you can use. For example, just simple transfection into cells. On the left here, these are plots of a cell line with a BMAL1 luciferase reporter, and the levels of bioluminescence are actually about 100 times brighter than HER2-LUC, but the rhythms are not as beautiful here on the left compared to the HER2-LUC on the right. Another important consideration for the reporters is the half-life. Depending on the dynamics of the process that you're studying, you have to make sure that the half-life is not too long of the reporter, because if it sticks around forever, you're not going to be able to capture the dynamics changes in the physiology very well. So, what we're measuring here is circadian, approximately 24-hour rhythms in gene expression, and you want the half-life to be considerably shorter than that. For the luciferase reaction, actually the effective half-life is pretty short already without doing anything special. It's about 1 to 4 hours, mainly because of product inhibition. So the luciferase is inhibited by its own reaction products, and even though the protein doesn't actually turn over quite that fast, the effective half-life is fairly short. For fluorescence though, you do have to worry about this, because the GFP half-life is about 26 hours. So what people normally do is to destabilize the GFP by adding a PAST sequence to increase the rate of turnover of GFP. And so I illustrated that in these movies down here from Nick Hastings' lab. This is a time-lapse movie of HER1 driving CFP without any destabilization. And so the half-life is so long that the brightness doesn't change very much, is the point. Whereas if you do a similar reporter, this is YFP instead of CFP, but the significant difference is that it's destabilized. It has this PAST sequence to destabilize it. And here you do see the getting brighter and dimmer and brighter and dimmer, the cycling, which is what you want to see. Onto luciferases. There are lots of different naturally occurring luciferases. Most of them are marine species. I won't say much about bacterial or marine luciferases because the bacterial one is not expressed in mammalian cells, not useful to us. And all the marine ones, while they may have their virtues, they all use the celanterasine substrate, which I detest really because it's expensive, it's insoluble, it's unstable, and there's some toxicity. So for those reasons, we use exclusively the beetle luciferases. The most commonly used one is an optimized version of the firefly luciferase, which is available from Promega. They've done some codon optimization, removing transcription binding sites to make it just cleaner than the wild type. I should note that you might expect this as blue-green. It's actually orange at 37 degrees at physiological pH, so that's something to keep in mind. There are other colors available from different beetle species that have been used for simultaneous monitoring of multiple parameters, which is kind of cool. And the reason we love these beetle luciferases, again, is because the substrate luciferin is very easy to work with. It's cheap, soluble, and non-toxic. There are beetle luciferases also from Toyobo in Japan. I won't say much about those. Let me move on to lenses, which are very critical. The whole trick in bioluminescence imaging is to try to optimize signal-to-noise ratio so that it's important that the lens be as bright as possible. And the brightness of a lens is determined by a few things. The numerical aperture is the measure of the cone of light, how big a cone of light is collected by the lens. T is the transmittance, or how of the photons that reach the lens, how many are transmitted through it. And something you may not be as familiar with is this concept of magnification being important, because what magnification really does is it spreads light over more pixels. And so the light reaching any individual pixel is greatly reduced the more the image is magnified, which is bad for bioluminescence. And for fluorescence, it's a little bit different because light goes, the incident light goes through the lens, and the light from the sample goes back through the lens in the opposite direction. So the light passes twice through the lens. For that reason, the numerical aperture is relatively more important than the magnification. And so that's why microscope reps tend to focus on numerical aperture exclusively, because they're much more familiar with the fluorescent probes. But all of these three parameters are important. For bioluminescence, actually, the best lenses usually end up being relatively low magnification lenses for the reason that I mentioned before. We like this Olympus XL Fluor, but there are a variety of lenses available from different manufacturers. This one, the Olympus XL Fluor, is the brightest that we found. It doesn't fit on a standard mount, requires a special slider. So it's a little bit inconvenient, but and then another alternative that some people have used is to use one of these other lenses, or even a higher magnification lens with a high NA, but then in a second stage have another lens that reduces the magnification afterwards using a demagnifying tube lens in series with the objective. So that's another way to get high NA, but the ultimate magnification is reduced. Say a little bit about cameras. Colin Coates is going to say, from Andor, is going to say something more. But with cameras, what you want to pay attention to is quantum efficiency, which is the proportion of incident photons that are detected. The read noise, which is the noise penalty that you get with each readout from the camera chip, and it's reduced by slow readout or binning, as I'll mention in a second. There's dark current to worry about, which is thermal electrons. It's reduced by cooling. And there's noise associated with this dark current, which increases with exposure duration. Now, Colin is going to go into more detail, but basically there are two approaches to cameras. Okay, the best detectors really are probably still CCDs, CCD cameras, charge-coupled devices that use the photoelectric effect. Photons dislodge electrons from a chip, and those electrons are detected by circuitry in the camera. It's a basic principle. But there are two approaches. One is the conventional CCDs, which are best for long exposures. And these generally have very good quantum efficiency. The drawback is that the read noise is fairly significant. So that means that for each exposure you take, there's a bit of a penalty in read noise. But they do have low dark current, and that allows long exposures. The other approach to get around this problem of read noise every time you take an exposure is taken with two types of cameras, either intensified CCDs or electron multiplication CCDs. These are best for short exposures because what they do is they amplify the signal above the level of read noise before they even get to the signal even gets to the CCD. They take different approaches to do that, which I won't go into here. But the intensified CCDs, they both have their problems. The intensified CCDs generally have a low quantum efficiency for the initial detector. They're complicated, expensive. The EM CCDs have their own special kinds of noise, clock-induced charge, and generally have high dark current. So there are pros and cons for these cameras. We generally, in our lab, we use exclusively the conventional CCDs. And so let me tell you about how to optimize this whole detection process. It's all about maximizing signal-to-noise ratio. You want to make more photons. You don't want to lose any. You don't want to have any light leaks because that creates noise. You want to buy a big lens, a good camera, and use the right settings. Okay. So at each of these stages, let me tell you something about optimizing. First, for the luciferase reaction, consider using a brighter reporter. As I mentioned, we use the knock-in reporter because it's very faithful to the circadian clock's gene expression. But if you're desperate, you can't, and the signal is too low, you could use a reporter with multiple copy number or consider using brighter luciferase. You want to buy good quality luciferin. Some cheap varieties have yellow inhibitory byproducts. We use the luciferin from Biosynth. To a degree, up to a certain limit, you can use more luciferin, which can get you more brightness. The enzyme saturates around 1 to 5 millimolar, though, and toxicity begins at about 2 millimolar. So we use 1 millimolar luciferin, which is just in the culture medium. Very important to have a dark room because stray light adds noise. So it's not, you might think you could just subtract out any extra light as background, but the real problem is that extra light from the background is a source of noise, which you really can't get rid of, except to prevent it in the first place. So you want to turn off any little sources of light in the room, the microscope, any little LEDs on equipment, the computer monitor. You want to cover the equipment LEDs. You want to drape the microscope in blackout cloth, and then sit there for about 15 or 20 minutes because your eye is actually a very sensitive detector of any stray light, and you'd be surprised what you can see after sitting in the dark for 15 or 20 minutes. You want to make sure the light path is clear because every photon counts. Consider emitting phenol red from the medium It's a pH indicator commonly used in cell culture, but of course it's colored, so that means it absorbs photons. It's actually not that big a deal in an inverted scope because you're looking through a pretty thin section of medium, culture medium. But in a luminometer, it's a big loss, or an upright scope. You want to make sure everything is clean, culture dish, lens, any other optical surfaces. We put the camera on the bottom port so that it doesn't have to bounce off of any mirrors, because you lose, every time the light goes through a filter or bounces off a mirror, you lose photons. Now, let's get down to the nitty-gritty here for how to use these cameras. The critical things are the quantum efficiency, which should be above 90% for most of the good cameras. The read noise for a conventional CCD should be about 2 or 3 electrons or better. The dark current should be less than 0.001 electrons per pixel per second. And so it's important to buy a good camera with these good specs, but it's also important to have the right settings. To minimize the read noise, you can set the camera to read out slowly. This reduces the amount of noise associated with this readout process. And that's very critical. It's not always obvious from the documentation, but it's critical. You want to minimize the dark current, and the way to do that is to cool the camera, the CCD, as much as possible. We cool to -90 Celsius. And finally, you want to actually minimize temporal and spatial resolution, because it's not about getting pretty pictures here. It's about collecting the most photons. So you want to use long exposures and something called on-chip binning, which is a way of combining the readout from multiple pixels on the camera as if they were one superpixel, and there's only one read noise penalty associated with that whole group of pixels that collect more photons than a single pixel. So you come out ahead in the signal to noise with on-chip binning. All right, and also low magnification minimizes spatial resolution too. So really the best way to optimize things is to collect as many photons as you can, and that's because a significant amount of the noise is what's called shot noise, which is just stochastic fluctuation of the signal itself. And that increases only as a square root of the number of photons, whereas the signal increases in direct proportion to the number of photons. So you're better off for signal to noise having more photons. And as I said, longer exposures or binning always improves the signal to noise ratio, although at the expense of temporal or spatial resolution. Now, for our studies, the circadian studies, we don't have to worry about the temporal resolution very much. Things don't change that fast. And spatial resolution, we only need enough to be able to distinguish one cell from another. So this is a still image of fibroblasts, Purkinje fibroblasts. And what I want you to notice is that it's not that pretty. It's got pixelation, so they're individual cells. So this is a cell, for example, right here. It overlaps a few different pixels, but we're not trying to make this a pretty picture by presenting excellent spatial resolution. We'd rather sacrifice spatial resolution and bin the pixels to maximize signal to noise, and that's so that we're deliberately sacrificing spatial resolution here is what I want to emphasize. Okay, I'm almost done. Just a couple more things. For image processing, the first thing we do is subtract a background image, which removes any kind of stable CCD defects, and then cosmic rays, and I'll say something about that in a second. Define cell regions, so we're studying individual cells in a whole field, measure the brightness in each region of interest, convert from arbitrary units of the camera to photons per second, and then standard things like measuring the period, phase, and amplitude of rhythms. So this is kind of the standard kind of data analysis that we do. Let me say something specific about cosmic rays, because this is of broader interest. So this is an SCN slice, and you'll notice that there are these little annoying dots around here, which are actually the result of extraterrestrial charged particles, which come in and hit the CCD chip. And so they're artifactual. You like to get rid of them, and you take advantage of the fact that they're randomly distributed. The reason that you see these in the first place is because with bioluminescence, you're using such long exposure times. Normally, with fluorescence, you would never see these. So this is something that's a kind of unique problem for bioluminescence because of the long exposures. But one 30-minute exposure and the next one are not going to have cosmic rays in the same place. So this is the first image here, and then I'll show you a second image, the next 30-minute exposure, and notice that these little dots are in different places, right? Did you see that? So what we can do is we can take advantage of that. We can construct artificially a compromise image that is the minimum. Each pixel is the minimum of the two corresponding pixels from two successive images. So it's like doing a running minimum algorithm, and the result is something very nice like that. You could also just filter for super bright things because the cosmic rays tend to be brighter than the actual sample. That's another way to do it. All right. So so that's what I have to say for today. I just wanted to mention that a lot of this technical development was done in Steve Kay's lab at Scripps Research Institute. The Per2Luck mice were developed by Seung-hee Yu in Joe Takahashi's lab at Northwestern and UT Southwestern. And in case you missed anything, I have a couple of review papers. The most recent one in Cold Spring Harbor Protocols is going to be made available by the organizers of the webinar. All right. So I will now turn things over to my colleague Colin Coates from Andor, and take it away, Colin.
Okay, thank you very much, David, for that. I will just give me 1 second to share my screen and go into presentation mode. Hopefully that's all working. Okay, so hello everybody. I'm going to spend the last 10-15 minutes of this time talking in more detail about the detector technologies. So first of all, I'll spend a little bit of time just again recapping what it means, or sorry, the concept of sensitivity and signal to noise, because it is an important thing to grasp in this application area especially. I'll say first of all what typically makes a good detector choice for fluorescence microscopy, and then I'll spend the bulk of the time then talking about contrasting that with how you would choose a detector for bioluminescence microscopy specifically, and also to an extent how you might even want to optimize some of those detectors for that as well. So what makes a detector sensitive? So if I break this down into very simple terms, the first thing which is often recognized if you go onto a specification sheet for a detector, for example, is the quantum efficiency. And that is, sorry, there's a little bit of noise. David, I wonder, would you mind just muting your headphones to see if that helps? That's helped. Thank you, David. Okay, so the quantum efficiency is it's typically shown then as a graph on a specification sheet for a camera. So it's the graph of wavelength versus the light detection effectiveness given as a percentage. It's basically how good the pixels are at capturing incident photons of any given wavelength. So that's usually a pretty obvious thing to spot on the spec sheet. And then the other thing to consider is what I would call the noise floor of the camera. And the analogy I'm using here is looking at an object sitting in grass. So if that grass is short, if we cut the grass, then it's much easier to see the object being the signal in this case. It's quite a good analogy for the noise floor. And then, so really what we're trying to do with a sensitive camera is make sure that each of these parameters are optimized. So if I then show you a pictorial view then, taking, say, a light intensity profile through an imaged object. What I'm trying to represent here at the bottom is the noise floor. Now that is further broken down into the read noise and the dark noise. So David's already mentioned this. The dark noise is really the noise associated with your dark signal. So you will see parameters in the spec sheets associated with each of these as well. I'll talk a little bit in more detail about how the read noise is affected by speed in another slide or two. But suffice to say for now, this is typically how the noise profile hangs together. You have your noise floor that you really want the object to be greater than in order to see it. And then you have this concept of shot noise variation, sometimes called Poisson noise as well. And that really describes your pixel to pixel or your frame to frame variation of the signal. So the more photons you have in the measurement, the more favorable your signal to shot noise ratio will be. And how too high a shot noise can be manifest in an image is that it will just look that bit more ill-resolved, that bit more grainy. So the longer you leave your exposures, if you have a very weak signal, as is typical for bioluminescence, then the longer you leave your exposures, or the more pixel binning has also been discussed, all sum together to increase your signal level and improve your signal to shot noise ratio as well. So that's an important factor. And of course, shot noise is also affected by the QE, again, just by way of the fact that a better QE, better quantum efficiency, collects more signal, and you get a better signal to shot noise ratio. So moving on, it's worth pointing out, because it was mentioned by David quite rightly, that fluorescence is hindered often by fluorescence background. Or photon background, and that often is the real detection limit of your experiment. In many setups, in many modalities of imaging, the shot noise associated with the photon background that you're unable to get rid of actually is the true detection limit. Here I'm showing an example from several years ago of a turf setup, and what I've done here is take a line intensity profile through a random part of the signal. It's actually single molecules isolated, and we're using turf to try and reduce the fluorescence background, and that generally is very effective. But even in this case, in a modality that is designed to limit the amount of photon background, we're still seeing a bunch of spikes in the background here. This is an EMCCD image, so this is single photon sensitive. If we see signal here, it's from photons, not from the camera noise, because we completely overcome the camera noise with an EMCCD. So here in this case, for example, I can see a large spike here is from a single molecule. I can see another one here from a single molecule, but all this forest of spikes in the middle, it's hard to pick out. Might there be another molecule in the middle here? We don't really know because there's so many photons scattered still from that background. So it's important in fluorescence microscopy always to make as much effort as you to limit the fluorescent or out of focus fluorescence background. Now, many applications, however, require speed. And when I say speed in this context, I'm talking frames per second rather than seconds per frame. So there's many techniques and types of microscopy setup, that several of which I'm listing here, like cell motility and typical fast calcium ion signaling, for example, in smooth muscles, that would need fluorescence. Simply, we do not have the luxury to be able to apply long enough exposures to make bioluminescence microscopy practical. So here we're talking about simply the driving need to use fluorescence microscopy. So then what technologies do we recommend? Oh, sorry, let me talk about this first. So CCDs up until a point a couple of years ago used to be pretty ubiquitous for fluorescence microscopy, and it's pretty much because they were the only show in town. But the big drawback of using CCDs for fluorescence microscopy is that when you actually want to go faster, when you want to push those frame rates into frames per second, you have to read out the CCD faster as well. So CCDs have sequential readout. All the pixels are read out in turn through common output amplifier, which is where the noise happens. The faster you read those pixels out, you get faster frame rates. The higher the noise becomes, the longer that grassy noise floor becomes effectively. And that's exactly what's happening in this view here. I'm showing exactly the same relative intensity scaling on the same cell, the same fluorescence image cell, but the difference is I'm reading out the top one at 1 megahertz, which is sufficient for about 1 frame per second, and the bottom one I'm reading at 20 megahertz pixel rate, which can get the CCD up to 11 frames per second. So it's going between 2.4 electrons noise on the top, and then I'm getting a longer grass on the bottom with 5.5 electrons, and you can see the difference in the contrasting capability then. The grassy noise is much more evident. Again, remembering this is the same relative intensity scaling, you can visually see the higher noise and therefore the reduced contrasting capability. So for fluorescence, that's not a great thing, because that means you're then left with the option, if you want to maintain the frame rate, your only option really then is to increase the excitation power, but of course that will result in an elevated rate of phototoxicity and photobleaching of the dyes as well. So CCDs, great for slow stuff, not so great for fast stuff. So that's why there have been alternate technologies that have come to the fore and come to bear over the past number of years. And I'm showing what I'm recommending here is really the two technology types that we recommend for fluorescence. On the left-hand side, I'm showing scientific CMOS. It's actually the newest technology type to enter the market. And I've written here it's a workhorse microscopy camera. It really is an all-singing, all-dancing technology, capable of so much, and generally will deal with the vast majority of fluorescence imaging challenges. So it's a great combination of specifications. It's very low read noise, even at fast speeds. It's typically down to 1 or 2 electrons read noise at speeds of full frame rates of up to 100 frames per second, while maintaining that very low noise is something that CCDs just can't do. Its quantum efficiency is getting there. We're now actually looking at CMOS sensors that have quantum efficiency around about 80% peak, and also very broad response across the wavelength range, very high dynamic range given the small pixels also. So it's generally a good workhorse camera technology. And then we have on the ultra-sensitive end of the scale, we have the EMCCDs. They've been around for 15 years now, and they are single photon sensitive, back illuminated CCDs, so they're very good quantum efficiency, and they're single photon sensitive, meaning every photon can basically appear as a signal. Nothing is lost in read noise because there practically is no read noise on an EMCCD. So these technologies are used when you're at the sort of low tens to single digits numbers of photons per pixels per frame. So they're used very typically in single molecule detection. They've been very widely used in super resolution microscopy, and also in modalities of fluorescence that are designed to reduce the amount of light coming from outside the focal plane, and which inherently makes them low light as well, such as spinning disk confocal microscopy and TIRF, which I've already mentioned. So I mentioned frame rates. The SCMOS technology, as it happens, is probably the best example of a detector technology which can be pushed, maintaining low noise to a very fast frame rate. And I'm giving some examples here of how our Xyla SCMOS can be pushed. If you then start to use double rays smaller than its 4 megapixel native format, you can get much faster frame rates, down to an extreme one here, which we call the fluorescence correlation spectroscopy mode, which is 2048 pixels wide by 8 rows high, which is now giving more than 26,000 frames per second. So that's the sense of what I mean when I say these cameras are good for fast frame rate applications. So let's move into bioluminescence microscopy, the main point of the talk. So right up front, I'll state the camera technologies I'm recommending for bioluminescence are slow scan back illuminated CCDs, and the example from Andor's range I'm giving here is the Icon M CCD, which is a 1 megapixel, 13 micron pixel size, so quite a decent photon collection area per pixel as well, to make sure you're not really wasting any photons with smaller pixels. Of all the technologies, it offers the lowest possible dark current, so we cool our cameras down to very deep cooling temperatures anyway to minimize dark current, but even with that, the sensors associated with these slow scan CCDs lend themselves to minimizing dark current to 1 or 2 orders of magnitude beyond any other detector technology, so they still remain fantastic for very, very long exposure applications, of which types of bioluminescence microscopy very much fall into that criteria. When, as I've mentioned, when you slow down the readout to, for example, sub-megahertz pixel readout speeds, you can still attain very low noise, around about between 2, 2.5, maybe up to 3 electrons. And they have very broad quantum efficiency curves, which can be important if you like to use a selection of different reporter tags. And then I'm still actually recommending EMCCDs for bioluminescence microscopy. So I've tagged this here as, in contrast somewhat to how I'm positioning the CCDs for long exposure times, I'm saying EMCCDs come into play when you want that bit more exposure flexibility. Even in bioluminescence, sometimes you can be driven to go down to seconds per exposure rather than minutes per exposure. And their EMCCDs can be used to give the better result, being single photon sensitive and not having to worry about the impact of the read noise floor in the measurement. So that's what I'm trying to get across here. They can be used for both long, short and moderately long exposures. And there's another thing I'll mention later, you can operate them in photon counting mode, which can have a distinct advantage for bioluminescence microscopy. Another interesting facet of EMCCDs, they're actually a CCD and an EMCCD in one. The two models I'll recommend have what's called a dual amplifier output, which means that through the software you can operate it as a CCD, or should you need to make it single photon sensitive for shorter acquisitions, you can put it into EMCCD mode. So that's quite good. You get a lot for your money in that sense. So focusing first of all on the ICON MCCD, what I'm showing here is the different types of sensors you can get in this model, but I'm drawing particular attention to this one, which we've labeled BEX2DD, bit of a mouthful, but it's actually got 3 different technologies working simultaneously to produce the broadest quantum efficiency curve that you can get for silicon-based cameras, basically. So it's giving great, very high quantum efficiency from around about 400 nanometers right up here to about 900 nanometers and then tailing off either side. That gives fantastic coverage, and that will cater for any combination of luminescent tags you could want, from luciferias right up to some Brett sensors, for example, that may be a luminescent reporter tagged to GFP or another fluorescent protein for enhanced brightness. So very broad QE curve there. I mentioned the noise already, down to sort of sub 3 electrons, down to even 2 electrons. They're cooled. Now we do cool our cameras very extensively. We've got a lot of expertise in this regard. We use vacuum cooling technology. When you just have the fan running, we can stably manage minus 80 degrees. Cooling of the sensor to minimize dark current. If you want a bit of extra boost to that, you can flow water through these taps instead, and we can push it down to minus 100 degrees. That gives us dark currents that are exceptionally low, so you can see the number of zeros in there for electrons per pixel per second. That translates into 0.36 electrons per pixel per hour. So when we're talking about bioluminescent exposure times that are 10, 20 minutes, we know here that the dark current is going to be way sub-electrons, so it's lower than the read noise. Basically, it's not a factor. That means we've eliminated the dark current, even under very long exposures, so that we don't care about it anymore. So, just an example from literature of one of our CCDs in use. This, again, I've chosen this example because it is a long exposure application. It's a time-lapse image of a protein tagged to luciferase from mice stem cells, mice embryo stem cells, and here we've been using 20-minute exposure times and still managing to probe the gene expression cycles involved. So moving on, being conscious of time. With electron multiplying CCDs, a little bit more detail just on what's happening here. It's not so much that we're reducing the read noise per se. What we're doing is we're amplifying the signal so much that the read noise doesn't matter anymore. We've effectively made that read noise negligible. So we haven't cut the grass, we've just amplified the object sitting in the grass. So they're single photon sensitive, have again these high and broad quantum efficiency curves, and if you need them to, because I've also recommended these cameras for fluorescence microscopy, at full frames, full resolution, you can push them typically between 30 and 60 frames per second. And that is just showing you what happens when you turn on the EM gain. This is a fluorescence measurement, but you can see the difference. The read noise is very dominant there. When you turn the gain off or the gain on, you amplify the signal so much that the read noise is scaled out. So I'm now scaling on the amplified signal, and the background is nice and black in contrast. So the two models I recommend of the Ixon EMCCD for bioluminescence are shown here. The one is called the Ultra 888, the other is called the Ultra 897. So the 888 is the largest field of view. You can see the representation of its relative field of view here compared to the 897. 13 micron pixels. It is the more expensive of the two. Typically, the more silicons involved in a sensor, the more you pay for it. The 897 is 512, 512, not as high resolution, but interestingly, it does have a larger pixel size, which can be favorable for weak bioluminescence. It may mean you can actually avoid binning, for example. The larger the pixel, the more photons it collects, and the greater your signal to noise overall. So that's two possible recommendations of EMCCDs. Both will do a very good job. Here I'm showing basically what happens again when you increase the gain. I like this representation because while it was done not actually using bioluminescence, it was done in a very controlled environment on a low light light-tight imaging rig. I was able to reduce the light intensities to a level where they were so weak they were depictive of a very, very, very weak signal. And I like it because it's actually got a zero photon background as well, or practically zero. So all I'm left with is the noise of the detector. So basically it's showing the read noise here. If I take a line profile through one of these very dim pinhole spots, I have the read noise. As I increase the gain, you can see the noise being amplified above the read noise, and the read noise is basically scaled out of existence. So that's again the concept of an EMCCD. It is useful to push EMCCDs to deeper cooling for longer exposures. Have to be quite careful with EMCCDs because the mechanism that amplifies the photon will also amplify a thermally induced electron as well. So it amplifies dark current. So for long exposure specifically, it can be quite crucial to make sure you are doing as much as you can to cool the camera as much as you can. So when you flow water through the water taps on our camera, you can push down to -95 and one of the models to -100, and that's a difference it can make. Again, this is an extraordinarily weak condition, very, very typical of a challenging low light luminescence experiment with a 2-minute exposure, and you can see going from even -70 to -95 the difference that can make in terms of further minimizing the dark current influence of an EMCCD. So just something to be aware of. Now, with EMCCDs, there's no such thing as a free lunch. The amplification mechanism that amplifies your signal above the noise floor does have a downside, and that's called multiplication noise, sometimes called noise factor in the literature. And it's well documented, well measured. It basically takes the shot noise of the signal that I talked about and amplifies it by a factor of √2. So it increases your relative shot noise. Interestingly, you can overcome this again doing single photon counting, and bioluminescence, you could argue, is one of the few techniques in biology where you actually may practically be able to implement single photon counting. So how do we go about this? Single photon counting is basically a series of binary images. In doing this with an EMCCD, you will deliberately set up really quite short exposures, maybe in the tens of milliseconds range, and record them quite fast. And the reason it suits bioluminescence is in bioluminescence there's a good probability that your signal, will be at the single photon level. So for single photon counting, you don't want to have, pixels carrying tens of photons or big groups of bunched together photons across, a radius of pixels. You want to have basically just a scattering of photons in any single given frame. But then what you're going to do is take lots of those short exposure frames and then basically do a thresholding technique. Just count the number of photons that appeared above a threshold that you've set. Then you'll have a series of binary images and you add them together. And that gives you your resultant photon counted image. And that actually gets you back into a situation where you don't have multiplication noise. It gets you back to your native signal to shot noise arrangement, if you like. So we're quite conscious of this in our software, for example. In our cameras, we have put the capability to do even either real-time photon counting or post-processing photon counting. I'm actually a fan of the latter. I would just take the raw data, and then afterwards I can make decisions on how many groups of images I want to bundle together to get a single output image. That means I can experiment with that. I can sort of group together, say, 20 images, groups of 20. That will give me a certain temporal resolution overall if I've recorded, say, 1,000 images of raw data. And if I don't like the signal to noise, I can say, okay, I'm going to sacrifice temporal resolution, and I'm going to group together 50 images at a time. I'll get better signal to noise, but I've sacrificed my overall temporal resolution, which may be acceptable. Just a point, there's another noise source in the MCCD, so clock-induced charge. We do a very good job of minimizing that, but just to illustrate here, for single photon counting especially, it is quite critical to minimize it. So what I'm showing here is a force condition of high clock-induced charge versus a low clock-induced charge of a series of added together photon counting images, and you can see the difference it makes in the contrasting capability of this very, very weak experiment. So just to finish off with a couple of slides, again not taking too much time, and I realize we're up on the hour, of where EMCCDs have been used for luminescence microscopy. Here's one example here, and the point behind this image really is that we're not talking about minutes per frame, we're talking about 2 seconds exposures for the bioluminescence. So actually bioluminescent 3D stacks were recorded here. Each image, actually there were a series of 5 focal planes detector, 5 slices across 3D, or sorry, across Z were recorded, and each slice was 2 seconds, so 10 seconds per 3D stack. And finally, this is an interesting one. This was an experiment which was involving BRET. So it's a bioluminescent resonance energy transfer where a quorum was tagged to GFP. And this is a mechanism to actually make the overall signal that bit brighter. If the energy of the quorum is transferred to GFP, actually you get a little bit of a brighter signal. That can lend itself then to slightly faster bioluminescent measurements, and here we're looking at calcium oscillations. Again, 2 seconds per exposure were used in this work. So you've heard a lot today about bioluminescence microscopy. Hopefully you've got something out of it, and we thank you very much for your attention.
Thank you, David, and thank you, Colin. Unfortunately, we do not have time for questions. However, we will be sending the presenters the questions that we took today and publishing the answers on our website, photonics.com. If you missed any part of today's presentation or want to share it with your colleagues, you will be able to find the video later today at photonics.com/webinars. Once again, thank you for attending today's webinar sponsored by Andor Technology. I'm Robin Riley for Photonics Media.